Hello and welcome to Lecture 7. We're going to pick up where Lecture 6 left off. So we covered vision. We're going to cover all the rest of the senses in this lecture. We're going to start off with the mechanical senses, which really include the auditory and vestibular systems as well as somatosensation, and then we'll go over the two chemical senses that we have, which would be taste and smell. All right, we'll start with auditory stimuli. We're going to decode the sound waves into electrical and chemical signals in our cochlea and then relay those to our temporal cortex in order to make sense of all those sounds. Sounds are really nothing more than just compressions of air that are going to travel as waves. Just like light travels as a wave, so does sound. And so the different wave properties are going to determine the different characteristics of the sound. For example, the loudness has everything to do with the amplitude of the waves. We measure sound with decibels or decibels, however you'd like to say it. 0 decibels would be the limit of human hearing. For every 10 decibels that you add on to that, it gets 10 times louder. So if a typical conversation is going to be at 60 decibels and a jackhammer's at 130 decibels, that's a 70 decibel difference. And thus it's 10 to the 7th power or 10 billion times louder than a typical conversation. The wavelength of sound will determine the pitch. So the shorter the wavelength, the higher the frequency, and the higher the pitch. And we'll see that this pitch is going to be encoded based on the region of the cochlea that gets stimulated. So different wavelengths of sound are going to act on different parts of the cochlea. And that in large part determines pitch for us. Okay, so first of all we have to review the outside of the ear here. We call that the pinna. It has this unique shape that's going to help draw sounds into the ear, and the purpose of the pinna is to kind of allow us to detect where sounds are coming from better, but then to also direct those sounds into our auditory canal so it can vibrate the tympanic membrane or the eardrum. So here's some sound waves coming into the ear there. They're going to move through the auditory canal and vibrate the eardrum. So that's the yellow membrane there, also called the tympanic membrane. Okay, so we've moved from the outside world into the pinna, into the auditory canal. Now we're vibrating the tympanic membrane. The vibrations of the tympanic membrane there are going to be transferred to these 3 bones called the ossicles, which will take the vibrations and essentially amplify them because the ossicles are going to get progressively smaller. So the malleus is connected to the tympanic membrane. Then the incus connects the malleus to the stapes, and the stapes is then connected to the oval window on the cochlea. Now the stapes is much smaller than the malleus, and so it essentially acts as an amplifier by taking the vibrations of the much larger tympanic membrane and transferring them without loss onto the much smaller oval window. Okay, so whenever we're vibrating that tympanic membrane, now shown in purple here, those bones also vibrate, and in doing so they're going to push and pull on the oval window, which is going to move a fluid in that yellow cochlea there. All right, the cochlea has a snail shape, but it's really just one continuous path that loops back. If we uncoil it, you can see that that's the case. Embedded inside the cochlea, we have our organ of Corti, which is actually where we're going to sense vibrations here. All right, so right now all we've done is had those ossicles vibrate the oval window on the cochlea, and that's moving around a fluid called perilymph in the cochlea. And as that fluid moves around, it starts to push and pull on different parts of the cochlea, which we can see at the bottom of this illustration. You can see part of that green portion, the basilar membrane there, is being pushed downward. Okay, so the vibrations of the perilymph that are going to occur, they're going to be sensed in the organ of Corti. You can see the organ of Corti shown in the bottom illustration. The organ of Corti there is then inside the cochlea. It's going to be connected to the basilar membrane, and it's going to be sandwiched between that and another membrane called the tectorial membrane. And within the organ of Corti there are different hair cells. So what's going on here is we have sound waves coming in, okay, and you can see in the top illustration there that different frequencies are going to stimulate different parts of the cochlea. And whenever we say stimulate different parts of the cochlea, what we're talking about in particular is causing movements of the basilar membrane that's going to affect these hair cells. And it's, in a sense, what they're going to do is simply move those little stereocilia on top. Now what you should notice here is that we have really two sets of hair cells. You can see that lone group of one, that'd be the inner hair cell, and then we have the group of three outer hair cells. Both of these are going to have stereocilia, and when the stereocilia are bent, that's going to then generate electrical activity within the hair cell, but they're going to respond differently. The inner hair cells are going to be responsible for transmitting that information to the auditory nerve. What we're going to find soon enough is that the outer hair cells actually change their shape and amplify the vibrations of the basilar membrane that are occurring. Within the organ of Corti, we're not surrounded by perilymph. Instead, we're surrounded by a different fluid called endolymph, which is going to be very high in potassium. So, if we can stop the movie for a moment and see this statically, at the top of this illustration, we have our resting cochlea, our resting organ of Corti. Nothing's going on. Once a sound reaches our ear, vibrates the tympanic membrane, vibrates the ossicles, vibrates the oval window, pushes the perilymph. That's going to push upward on the basilar membrane, as you can see in the bottom of this illustration. That in turn is going to push the hair cells up against the tectorial membrane on top. The outer hair cells are going to amplify this movement, as we'll see, and the inner hair cell is going to relay this information to the auditory nerve. The way that this occurs is through the movement of those little hairs on top, the little stereocilia. Stereo just deals with sound, and cilia deals with the fact that these are little tiny cilia, little hair-like projections on cells. That's all a cilium is. Now these are stiff, so when they get pushed, they don't bend. They instead just deflect in one direction, which pulls on the neighboring hair cell because they're actually connected to one another by what we call tip links. So if you look in the upper left-hand portion of this illustration, you can see 1, 2, 3 stereocilia, and there's a very fine tip link connecting them. This is just a filamentous protein in the cadherin group that's connecting one cilium to its neighbor. It's joined at these locations that we call insertional plaques. You can see that the tip link is connecting to the side of one stereocilium and then to the top of the other. Now what it's actually connected to is a trip channel, a transient receptor potential channel. These are mechanically gated ion channels. Now if you look in the upper right hand portion of this illustration, you'll see when the hair cells get deflected and that tip link gets pulled, it's actually pulling open the trip channel on the neighboring stereocilium. So by deflecting the hair cells, we can either create depolarization or we can cause hyperpolarization. And this is shown on the bottom portion of this illustration. We have a resting hair cell. Nothing is going on. It hasn't been pushed one way or the other. As the basilar membrane gets pushed upward, this is going to cause depolarization by pushing those stereocilia in such a way that it pulls on the tip link. And when that tip link pulls on the trip channel, that allows potassium and calcium to enter. Now in this case, potassium is actually depolarizing because endolymph is a very high potassium solution, and that shifts its reversal potential away from the very, very negative value that it is in resting neurons. Resting neurons are not surrounded by endolymph. Instead, they're surrounded by cerebrospinal fluid, which is low potassium. So that difference causes potassium to be excitatory in this case. Either way, when you push on the hair cells and their stereocilia deflect, in this illustration to the right or toward the longest stereocilium, that pulls on the tip link, and we have depolarization. On the other hand, when the basilar membrane moves downward, then the tip link, which has some resistance in it, actually pulls the stereocilia toward the shortest one, thus closing the trip channels, which are normally open a very small amount, and this actually causes hyperpolarization. So the vibrations of the membrane are going to create this oscillatory increase and decrease in membrane potential by moving the stereocilia around. Now, the reason that these hair cells have their stereocilia moved in the first place is because of that second membrane. Okay, without the two membranes there, without the basilar membrane that moves and the tectorial membrane that doesn't move, we wouldn't have deflection of the stereocilia. Okay, so we've sensed sound with the tympanic membrane. That moves the ossicles, that moves the oval window, that moves the perilymph, which pushes up on the basilar membrane. That deflects the stereocilia, which opens up trip channels and allows the potassium and calcium in the endolymph to flow into the hair cells, thus causing depolarization. As they're moving back and forth, we get these oscillatory changes in membrane potential. Now, the inner hair cells are going to then release a neurotransmitter. The outer hair cells, though, are going to amplify this movement. All right, and the way that they do that is by changing their length in a voltage-dependent manner. This plot shown on the right is showing you the relationship between membrane potential, shown on the x-axis, and the length of the outer hair cell, shown on the y-axis. Here they're showing you the change in length. So at the resting potential, shown there at the 0, 0 coordinates, you can see that this is the so-called normal length. As the outer hair cell depolarizes, or as we move to the right on the x-axis, you can see that the hair cell becomes shorter. Okay, so depolarization causes the hair cell to become shorter. Let's look at this illustration again. We've gone back one slide here. When the basilar membrane pushes upward, those outer hair cells on the right, they get depolarized, and when they're depolarized, they shorten. Thus, they're going to pull the basilar membrane up even further. Okay, so here they're amplifying those vibrations by changing their length. And the reason that they can do this is because of a very odd motor protein that they express called prestin. What prestin does is change its length based on the presence of anions, so chloride in this case. So whether the cell is hyperpolarized, where there's a lot of negative potential, or there's a lot of positive potential, that's going to determine the shape of prestin. So when the cell is hyperpolarized, that means that there's these negative charges that can come in and embed into prestin. When they do this, this causes lengthening. Thus, when the cell is depolarized and there aren't negative charges embedded in prestin, prestin shortens, causing the cell to shorten. And that's what we're looking at in the bottom of this. That's an isolated outer hair cell connected to a pipette on the bottom. And they're using that pipette to control the membrane potential of this hair cell. And what you'll see that it's actually lengthening and shortening. It's changing its length. And the only thing that's causing this is the change in membrane potential. And that's why the outer hair cells there are going to act as an amplifier. We'll call them the cochlear amplifier. So once we have some deflection of the basilar membrane and the outer hair cells are pushed into the tectorial membrane, that initial depolarization that's caused from the perilymph pushing upward on the basilar membrane, that's going to cause the outer hair cells then to actually push and pull on the tectorial membrane to move around. And so they're going to amplify these movements here. The inner hair cells are going to be even further depolarized because of these movements, and as such they're going to release more glutamate onto the auditory nerve there. Alright, now once we've released glutamate onto the auditory nerve, we have to get it up into our cortex. So the pathway there is going to be sort of multifold here. So we're starting at the bottom right. That's our auditory nerve. Some information will be sent to the superior olive to help us localize sounds quickly. There will also be a stop on the way in the midbrain where we hit the inferior colliculus. Don't worry about that. Where is it? That's an animation that I've clearly screwed up. This is going to allow us to carry out some reflexive behaviors. So if an object is there's a loud noise, we can have a startle response before we really perceive the loud noise. From there, we move on up into the thalamus, into the medial geniculate nucleus rather than the lateral geniculate nucleus that we touched on with vision. And then from the thalamus, we're going to head on over to the auditory cortex. From there, we're going to then head on out to the associational cortices to determine where sounds are and what they are. So the primary auditory cortex is broken up into the core, and then the belt and parabelt. The primary would be the core, and then the associational portion of the auditory cortex would be the belt and parabelt. Outside of the temporal lobes, we can determine what objects are by communicating with the frontal lobes. We determine where they are by communicating with the parietal lobes. This is kind of like what we do with our visual information, where there's a dorsal and a ventral stream. The dorsal stream up to the parietal lobe helps us determine where objects are that we're seeing. It does the same thing for sounds as well. Now we will figure out different components of sound, like pitch, based on the regions of the cochlea that get activated. This is where our pitch detection begins. Okay, so here we're back in the ear. On the left, we have our tectorial membrane that vibrates back and forth. We have our ossicles in the middle ear, and then those push on the oval window to help move perilymph around in the cochlea. So here we've straightened out the cochlea. And what you can see is the frequency map. Higher frequency sounds, which have a short wavelength, are going to vibrate the basilar membrane much closer to the oval window. Lower frequency sounds, which of course have a much longer wavelength, thus vibrate the portions of the basilar membrane that are further away. So the wavelength of sound determines essentially how far it stimulates on the basilar membrane. Okay, so we can see that in cartoon format here. Here's the actual data from some early experiments showing this. So the distance from the base, that'd be how far you are from the base of the cochlea, and then the frequency is shown on the y-axis. So the lowest frequencies are shown on the right here. Now the base would be the end of the cochlea, so as we're moving down in percent, so the base, if we look here, the base is going to be considered the oval window. Okay, so on the left, we're at the base, and as we move further to the right, we're moving away from it. So the top, 100% distance from the base, that means we're at the apex of the cochlea there. So we're the furthest we can be, and here we're detecting the lowest frequency sound. The highest frequency sounds get detected much closer to the base of the cochlea. And this is what we call essentially the place code. Depending on the place that's stimulated in the cochlea, that's going to distinguish different pitches for the most part. So it creates what we call a tonographic map, and this is preserved in the primary auditory cortex. So as we're moving through those different pathways, this relationship between the frequency of sound and its location is going to be maintained, not just in the cochlea, but also in our primary auditory cortex. And this works very well for frequencies above 500 hertz. So for most sounds that we're going to hear, the temporal code is going to be used for the very lowest pitches. So anything below 500 hertz, we're going to determine that pitch based on the frequency of action potentials. So anything that's below 500 hertz, it doesn't have the place code that we can see in this illustration where the location determines pitch. Instead, it's going to be the frequency of activity. So the the lower you go below 500 hertz, the slower the auditory nerve is going to be stimulated. So we use both a place code, which is going to work for most frequencies, and then we use a temporal code for the lowest frequencies in order to relay pitch from our cochlea up to our cortex. These hair cells that we've discussed are very, very important. Without them, we don't transduce sound into electrical activity, and if we don't do that, then we can't transform it into chemical activity, and we can never relay the information to our brain. Prolonged exposure to high sounds will kill off these hair cells. We can see an electron micrograph of normal hair cells shown on top. So we have our row of inner hair cells on the left there, and then the outer hair cells on the right. A damaged cochlea, which was exposed to very, very high sounds, you can see is lacking those uniform rows of hair cells. Without hair cells, no hearing. We can get around this, though. We can get around the loss of hair cells by using a cochlear implant. So we can just bypass those hair cells, and we can detect the vibrations of the cochlea using this implant here. So you can see the external portion here. This picks up sounds, and then it's going to directly stimulate the auditory nerve at different portions in the cochlea. So we're going to bypass everything here. We have an external sound processor that is going to pick up the different frequencies that are present in the sound and then relay that using the place and temporal code. So that external component is going to pick up sound. It's going to then relay that to an internal implant that's actually embedded in the cochlea. You can see here in portion 3, it looks like. That internal implant is going to receive signals from the outside, and based on the characteristic pitches that are determined, it's going to stimulate different portions of the cortex at different frequencies. And by doing this, we can then restore hearing even after we lose our hair cells. Okay, so we can use a few different cues to determine the direction of sound. We have two sides of our head, and as such, sounds that are coming from different sides of our head are going to hit the different ears at different times and with slightly different intensities. So in this illustration, we have a floating head of a man, and there's a sound source to his left. So in this case, the left ear is going to perceive the sounds as being louder than the right ear. Okay, this determination is going to be made in that lateral superior olive. There's also going to be a difference in the timing. So the left ear should encounter the sounds a little sooner than the right, right? Because the sound still has to travel that additional distance to the right ear. And this determination is going to be made in the medial superior olive. The model that we have for this is that different portions of the olive are going to receive input from the left and right ear at different times. And so depending on which neurons get stimulated by the input from the left ear and the right ear, that will determine whether or not the sound is in sync and thus directly in front or behind you, or whether it's further to your left or right. Okay, so in this model, what we're looking at are the dendrites of 5 different coincidence detecting neurons in the medial olive. There's input from the left shown on top and input from the right ear shown on the bottom. Now, if both if the left and right ear receive the sound at the same time, then they're going to stimulate the first neuron. So the left ear will stimulate the neuron on the left. The right ear will stimulate the neuron on the right. These will be out of sync. Then next, the second and fourth will be stimulated, and then the third. That third neuron is going to say they arrived at the same time. On the other hand, if the left ear was exposed to that sound just a bit before the right, the left ear is going to first stimulate the first medial olive neuron. The right ear hasn't done anything yet. Then the left ear will stimulate the second. Let's say finally the right ear receives input. So while the left ear is stimulating the third neuron there, only now has the right ear started to stimulate the fifth, and then they both meet up at neuron 4. Since neuron 4 was activated rather than the neuron 3 in the middle, that's telling us the left ear must have been activated first, right? Because it got further down the line than the right ear did. All right, so different parts of the superior olive are going to help us localize sounds using a few different schemes, either the volume or the timing. Okay, we're going to stay in the ear. We covered the cochlea. We're going to cover a different part of our inner ear now. We're going to talk about the vestibular system. And this is what allows us to track the movements of our head and which way is up. All right, we'll start with head movements. Head movements are going to be tracked by the semicircular canals. All right, we can see our cochlea in the upper left-hand portion of this illustration. That's going to pick up sounds, but the rest of our inner ear is going to pick up movement and gravity. So the semicircular canals are going to be our focus here. There are 3 of them, and they're going to track our head movements essentially in the X, Y, and Z planes. They're going to detect angular motion, so the one canal, and the anterior canal, will detect when our head is moving in a nodding fashion. Okay, so straight ahead. If you're nodding your head, then you're going to be stimulating your anterior canals. There's another canal, the posterior canal, that's going to determine when you're tilting your head from left to right. So if you were to try to put your ear to your shoulder, you're going to be stimulating your posterior canal. And then if you shake your head, as in replying no, then you're going to activate the horizontal canal. And depending on the activation of these 3 different canals all together, we can track how our head moves in all 3 planes. Now the way that we track this is by this specialized structure called the ampulla. Each semicircular canal contains an ampulla, and this is the motion detector. What we're going to find here is that when we move our head, the ampulla is going to move relative to a fluid in it. That fluid is going to then push on the cupula and bend the stereocilia. It's exactly like how we pick up sound, except it's not sound waves that are moving a fluid. It's the movement of our head that moves a fluid. Then we're going to move that large cupula structure that has the hair cells embedded in it. When the cupula moves, we generate electrical activity. These hair cells will then communicate not to the auditory nerve, but instead to the vestibular portion. Okay, it's going to eventually meet up with the so-called auditory nerve. Some people call it the auditory nerve. It's much better to call it the vestibular cochlear nerve because it relays information from our vestibular system as well as the cochlea. Okay, let's see a little animation of this in action.
Behind the vestibule is the third portion of the bony labyrinth, known as the semicircular canals. The canals project from the posterior region of the vestibule and are responsible for the detection of head motion in 3 spatial planes. The anterior duct senses forward and backward motion. The posterior duct detects moving up and down. The lateral duct senses moving left to right. Each canal contains a membranous semicircular duct where angular momentum is sensed. At the base of each duct is an expansion called the ampulla. Within the ampulla, long stereocilia of hair cells are embedded in the cupula, which sticks out into the endolymph. When your head moves, the endolymph moves the cupula and stimulates the stereocilia.
The vestibular system is also going to help us keep track of which way is up. Okay, so we're essentially in the same area, but now we're in the vestibule. That's the area between the semicircular canals and the cochlea. Here we're going to have these little calcium carbonate stones called otoliths or otoliths, depending on how you want to pronounce it. These are embedded in this sort of gelatinous matrix, and because the otoliths have weight, they're going to be pulled down by gravity. And when they're pulled down, they move the gelatinous structure and they bend stereocilia on hair cells. So depending on which way down is, that's going to determine which way the otoliths are moving. All right, here we have another animation that we can watch for this that might help it make a bit more sense.
The central, egg-shaped cavity is the vestibule, which contains a pair of membranous sacs, the saccule and the utricle. Inside the utricle and saccule are hair cells similar to those in the organ of Corti. The hairs are clustered in the macula, where their processes are embedded in a gelatinous mass and lie under a thin layer of crystals called otoliths. When the head tilts, gravity moves the crystal mass and distorts the stereocilia of the hair cells. This is how the saccule and utricle provide information about position with respect to gravity.
Okay, so once we've tracked the motions of our head and which way is up or down, we then relay this information, for the most part, to the brainstem. Because the vestibular system is mostly involved in sort of automatic or reflexive behaviors. It just helps us sort of perform some actions you wouldn't want to have to think about all the time. And so as you move your head, you want your eyes to also move accordingly so you can continue to look at the same object. So the vestibular nerve is really only a small portion of the vestibular cochlear nerve. So it's the same nerve that brings information about the vestibular system and the auditory information from the cochlea. So the vestibular portion of this will project to the vestibular nuclei in the brainstem, so in the medulla, and then it will head on to other brainstem nuclei that are going to control our neck muscles, that are going to help keep our head upright. So if it starts to dip one way or the other, we can keep the head pointed upward because we'll sense those changes in which way down is relative to our head. The brainstem nuclei will also control some eye muscles that will help keep our visual field constant. And so this information is of course going to be checked against other senses as well, so that as we're moving, so if we move our head, our visual field should change too. If our vestibular system is detecting that there's bouncing going on, and we look around, we should see the world bouncing. Sometimes the vestibular system and the other senses don't match up. For example, if you were to take a bus ride and read during the ride, you wouldn't see the changes that your vestibular system would feel, and sometimes this can lead to motion sickness. So if our vestibular system is picking up changes that our other senses aren't, this is going to be determined as an error signal. Something may very well be wrong with us, and so the first line of defense is to get rid of whatever you've eaten. It may be that you've eaten something bad that's causing some improper signaling to occur, and so to prevent potential toxicity, the vestibular system then stimulates that regurgitation. So you want your vestibular system to line up with your other senses. So an easy way to cure that motion sickness is to just look out the window. Just let your senses line up so that what your vestibular system is feeling, your visual system is going to agree with that information. Okay, the last portion of mechanical sensation is here. This is the somatosensory system. So this is detection of input to our body, so our sense of touch, as well as pain. All right, so here we're going to be in the dorsal root ganglion, and well, for the most part, except for the facial muscles, that'll be the trigeminal ganglia. But for our body, the dorsal root ganglion is going to have our somatosensory neurons, and these actually don't have any dendrites. They're what we call pseudo-unipolar cells, and they have a single process, which really is an axon, but it will function as the dendrite. It doesn't need to receive any chemical signals. Instead, it's going to receive mechanical signals. It's going to have some sort of stimulus sensing cation channel, and we'll go through some different receptors in a moment. That's going to create what we call a generator potential. You can see that in the left-hand portion of this illustration. So the receptor is going to create this depolarization. That depolarization is, of course, going to then drive action potentials. The frequency of these action potentials will depend upon the degree of depolarization. Then those action potentials will cause the release of neurotransmitter in the spinal cord. Okay, so the somatosensory neurons, don't think of these as your typical neuron. They have their cell body in the dorsal root ganglion, and then they send an axon that bifurcates to act both as dendrites and axon. Speaking of axons, we should be aware that not all axons are built the same. So the axons that carry out motor signals, those are the largest axons. As such, they have the greatest conduction velocity, because when you want to move, you want to move. Slightly smaller than that, we have our somatosensory neurons, the ones that are going to relay touch information, not pain. So non-painful information is going to be relayed on our group 2 afferents, which are a little bit smaller. They're still myelinated, but their conduction velocity is going to be reduced a bit. Pain is going to be sent along 2 different types of fibers. We have the small but still myelinated A delta or group 3 fibers, and then we have our group 4 or C fibers, which are very small and not myelinated. As such, their conduction velocity is the slowest. This is why when you hurt yourself, you'll first realize that you've either bumped something or touched something hot, and then you will feel the pain. Okay, pain follows sensation, and it's because of the size of the axons and whether or not they're myelinated. Okay, before we talk about pain, let's talk about good old somatosensation. We have a few different types of receptors that are really just these non-neural cells that are going to be surrounded by those A-beta afferents. So the second biggest afferents are going to wrap around these different receptors. There are some receptors that are very much near the surface. Those would be the Merkel's discs. These are going to be in the epidermis, and they'll sense gentle deformation of the skin. As we move on down, we'll hit the Meissner corpuscle. These are near the epidermis. They're going to be sensitive to low frequency vibrations. A bit below that in some species. So if you were a cat, you would have a Ruffini corpuscle or not. So we don't. But some species have these, and they are sensitive to skin stretch. We do certainly have Pacinian corpuscles, which are going to be the deepest and thus require the highest frequency of vibration to be picked up. Now, just as we have different receptors, there are also differences in the axons that conduct this information. Some of them are what we call slow adapting. If you look at the illustration on the right, on top you'll see a slow adapting afferent. So we see the skin pressure shown on the bottom. You can see an increase in skin pressure, and then it's dropped off. The slow adapting afferents fire very quickly, and then they will slow down eventually. Rapid adapting afferents will fire very quickly, and then they will very rapidly adapt to the skin pressure and stop firing. They'll only be sensitive to changes in skin pressure, whether it's an increase or decrease. That causes them to fire, and that's why you'll find your rapid adapting afferents mostly responding to vibration of some sort. So you can see here the Merkel's discs. Those are going to be near the outside of the skin. In the epidermis. They're innervated by slow adapting afferents. And so they can sense that gentle deformation the entire time that it's present because of that slow adaptation. You also find this in the Ruffini corpuscles. They're also innervated by these slow adapting afferents, and so skin stretch or sustained pressure will keep them active. The Meissner corpuscle, this is going to require vibration. Usually the movement of something along the skin, that'll cause little vibrations of the skin because we have rapid adapting afferents. The Pacinian corpuscle, also rapid adapting. As such, it's going to need vibrations of the skin, and because it's so deep, we need very high frequency vibrations to reach them. Once we stimulate the receptor, no matter which receptor it is, the A beta afferent is going to be activated in the same way. We just have a stretch receptor. So as we push on the skin, that pushes on the receptors, and that causes the stretching of the A beta afferent. So it goes from that closed, shown on the left, to the open conformation because of stretch. We're simply forcing open these receptors. That's all it is. Nociception, on the other hand, is a little different. Here it's not just mechanical stretch. Okay, the nociceptors are going to have bare nerve endings, so they're not going to be surrounded by specialized receptors. Instead, they have specialized proteinaceous receptors on their bare nerve endings. Some of them are going to have a few different trip channels, so transient receptor potential, that are going to be activated by acid or by differing degrees of heat. So a low heat will stimulate the trip V1, a much higher heat will stimulate trip V2, so they'll relay different information there. Some of these channels are going to sense acid, so if there's an increase in the amount of acid nearby, that's going to stimulate these sodium channels and thus cause depolarization. And then others are going to detect kind of a combination of these. Some will detect cold. Some are going to detect intracellular components to tell us that there's been tissue damage. And then they're going to relay this information to the central nervous system using, of course, those two different fibers that we mentioned early on. There's the A delta and the C fibers. They're both small. A delta is myelinated, so it's going to conduct its information faster than the C fiber, so it creates what we call the first pain. That's the initial sensation. The second pain is going to be the prolonged throbbing pain. Okay. Both of these are going to come on after we actually receive our somatosensory input, and they're going to be prolonged signals. Now, because these axons conduct the signal slowly, it arrives slowly, but it hangs around a bit longer. And that's going to help us remember this and be less likely to repeat that painful mistake that we made. The neurons in the dorsal root ganglia are going to project along a couple different pathways to reach the central nervous system. So just regular old touch information is shown in the blue there. That DRG neuron is going to first project up the dorsal columns, staying on the same side. Then it's going to cross over at the medial lemniscus, and then it will be on the contralateral side. It'll head on up to the ventral posterior nucleus in the thalamus, and then it will head on over to the primary somatosensory cortex. The primary and secondary somatosensory cortices are going to have several different body maps for different types of sensations. So this gentle skin deformation, the skin stretch, vibration of the skin, the length of muscles and joint angles, all of that information is going to get its own map. So there's at least 5 body maps out there. So this helps us know what's going on in our skin, where our sort of how our body is laid out, because if you were to know all of the muscle lengths and the joint angles, you could work out the position of all the parts of your body. The red pathway shown here is showing you pain. This is conveyed along the spinal thalamic tract. So first it heads on over to the contralateral side of the body, then it heads up the spinal thalamic tract to hit the same ventral posterior nucleus in the thalamus. The purpose of pain is to create a memorable sensation so that we prevent future harm. That's the purpose. So it's going to be conveyed up that spinal thalamic tract we just saw. The intensity and location will be determined to some degree in the somatosensory cortices. The negative or the unpleasant feelings are going to be constructed in other parts of the brain. So the somatosensory cortex receives information from the large somatosensory neurons, but also from the small pain nociceptive neurons. That'll help us know which part of my body has been hurt. But that long-lived ow feeling is going to be created elsewhere. One of the main players in that unpleasant feeling would be the anterior cingulate cortex. There are others. There's the insular cortex that was mentioned in the text. This will communicate to the amygdala to also create negative emotions. But the anterior cingulate cortex seems particularly important in creating the unpleasant feeling associated with pain. The more painful a stimulus feels to subjects, the more activity we tend to see in the anterior cingulate cortex. And that data is shown on the right there. You can see it's a fairly weak correlation, but it's fairly clear. The unpleasantness rating is shown on the x-axis, so on the left, not unpleasant. On the right, very, very unpleasant. And what we're looking at in the y-axis would be the bold signal in the anterior cingulate cortex, so the amount of blood oxygenation that we can detect with fMRI. So again, as you have more activity, you should expect greater blood flow to that brain region. And so we see more blood flow in the anterior cingulate whenever we have more unpleasant feelings from the pain. People who have damaged anterior cingulate cortices have reduced negative responses to pain. Okay, so you put these two bits of information together, and it's fairly clear that the anterior cingulate cortex is a key player in creating those unpleasant feelings that we get when we experience pain. Now, of course, we don't always experience the same degree of pain. We are able to adjust how much pain we feel, and part of that has to do with the activity of enkephalins, which act on opiate receptors. You can also take opiates to activate this system, and the opiates that people take as painkillers, for example, morphine, these are going to act in the midbrain and the spine to reduce pain. Okay, all analgesia means is a reduction or a lack of pain. Okay, so if you look on the top, you can see the model, the working model that we have for how opiates function. And what opiates do is inhibit this local inhibitory interneuron in the periaqueductal gray matter. That's the part on the left on that cartoon shown at the top. So the opiates inhibit that inhibitory neuron, and that allows that little green projection neuron to then become active. It communicates indirectly to the spinal cord, where it's going to then activate these inhibitory interneurons, which will reduce the activity of the nearby nociceptive neurons. So opiates, by inhibiting that local circuit neuron in the periaqueductal gray, are going to stimulate that analgesic pathway where we activate inhibitory neurons within the spinal cord. The opiates will also act on opiate receptors on the nociceptive neurons themselves to reduce their ability to communicate at that synapse. So not only are we activating inhibitory input to the nociceptors, we're also reducing their excitatory output. Now, of course, you can psych yourself into feeling this analgesia. This is what we call the placebo effect. People are certainly susceptible to this, particularly with pain, because we're able to drown out pain in case we need to work through some important or life-determining task. We can sort of put pain behind us. And part of the reason we can do that is because of the pathway that's shown on top. We have this built-in painkiller system. We just simply need to activate it. And so those inhibitory neurons that are going to be inhibited by by opiates, might they be tuned by other areas of the brain? And perhaps that's the case. So one of the main players here would be the dorsolateral prefrontal cortex. This is an area of the brain that kind of helps us create expectations and plans. And so if we expect a little pill that we take to cause some sort of pain relief, then the dorsolateral prefrontal cortex will become active and communicate to the periaqueductal gray matter to relay this expectation. We should feel less pain, and that expectation itself is enough to activate the periaqueductal gray and produce the analgesia. And this is why it's always important to have a placebo control. On occasion, we're going to have our own built-in systems that mimic whatever a drug is doing, and without that control, it's hard to separate what is natural and what is caused by the drug. Okay, I want to quickly run through our chemical detectors. So that would be taste and smell. We're going to start at the tongue. Tastes are going to be picked up by either G protein coupled receptors or ionotropic receptors. Okay, we've all heard of taste buds. Taste buds are going to have taste cells that respond to all 5 tastes. So each taste cell responds to one, but each taste bud responds to all 5 tastes because it has a mixture of these different taste cells. In this cartoon, we're having bitter as dark blue, and salty is a light blue, sweet is red. Umami is yellow, and sour is green. And so within each taste bud, which is shown on the left, you can see that there are taste cells that will pick up all 5 of these tastes. Now their organization, you can see on the right here, there's 3 different types of taste buds, and depending on the location of the tongue, you'll have different types of taste buds. But all the taste buds are going to have the same general features. They're going to have a little hole called the taste pore, where the chemicals in our food can diffuse in and interact with taste cells. Now each of these taste buds is going to have 50 to 100 or so taste cells that are going to detect all the different flavors. So at the top there we can see that taste pore, chemicals diffuse on in, and then it can interact with taste cells. Those taste cells are going to have little microvilli. These are smaller than the cilia, but they're little finger-like projections, very similar to the stereocilia that we've seen already. And those little microvilli are going to be littered with taste receptors. They'll pick up the taste. This will stimulate the taste cell, and then it will communicate to the primary gustatory nerve fibers shown at the bottom of this illustration. So the top is showing you the very top of our tongue. There's the taste pore. Taste will come in, interact with taste cells at the microvilli. If the taste cell is active, it's going to spit out neurotransmitter onto that yellow nerve fiber shown on the bottom. The neurotransmitter used here will be ATP. It's going to activate some purinergic receptors. So if you were to knock out those purinergic receptors, the P2X, then you're going to have severely impaired tastes, and that's what we see here. Here we're looking at recordings from the primary gustatory nerve in response to a few different stimulants. So a wild-type mouse is shown on the left, and on the right we're seeing that purinergic receptor knockout. So in response to sodium chloride, that's salty, you see that a wild-type mouse shows clear activation of its primary gustatory nerve. Okay, you can see that depolarization being recorded. The knockout mouse, nothing. Sucrose, right, which should be sweet, we also get a response. We get nothing in the knockout. As you move through the rest of the flavors, bitter, sour, and umami, you get a response in the wild type mouse, but nothing in the knockout. So they have no taste, but their tongue still is able to communicate mechanically. So if you were to touch the tongue, you can see on the bottom you still get a response, or if you were to drop cool saliva, that temperature can still be detected. So we haven't lost the ability of our tongue to feel. It can still feel cool, it can still feel touch, it just can't taste. Okay, so it's all about those purinergic receptors to taste. So we have 5 different taste cells. That means we need at least 5 different receptors, and that's pretty much what we have. So there's 5 different tastes. There's umami, that's going to be the savory or meaty flavor. There's sweet, there's bitter, sour, and salty. Those 5 flavors are going to, or I'm sorry, those 5 tastes are going to contribute to the flavor that we detect in our foods. So those microvilli are going to have a variety of different receptors. So if you are a taste cell that responds to umami, you're going to express the taste receptor 1 family of G protein coupled receptors. You'll also do that if you're a sweet sensing taste cell. They have different isoforms, so these receptors have to dimerize. There's at least 3 different members of this family, and so by forming different dimers, you're either going to bind to amino acids, mostly glutamate for humans, and that will allow you to sense umami or meatiness. So you're a G protein coupled receptor that binds to amino acids. G protein coupled receptors that bind to carbohydrates, on the other hand, are going to allow you to detect sweet. It's the same family of G protein coupled receptors, but it's different versions. Taste receptor 2, G protein coupled receptors, detect bitter. They're still G protein coupled receptors. So in the end, they're metabotropic receptors. They bind to either sweet, bitter, or umami signals, and then they're going to activate phospholipase C. So they're going to be GQ coupled, G protein coupled receptors. That's going to increase intracellular calcium through IP3 receptors, but you don't really need to know any of this. Okay, you just simply need to know that G protein coupled receptors bind to either amino acids, carbohydrates, or bitter compounds, and then stimulate the taste cell by increasing calcium levels. Sour and salty are detected by ionotropic receptors. Sour, which is really just acid, is going to be detected by different trip channels that are going to just allow the acid to move into the taste cell and depolarize it directly. This is very similar to how salt is detected. There's essentially a sodium leak channel that allows the sodium in sodium chloride to just directly move into the taste cell and depolarize it. So whether you have a G protein coupled receptor or an ionotropic receptor causing depolarization, in the end, what you have is an increase in intracellular calcium levels that then leads to the release of ATP to stimulate the gustatory nerve. Because each taste cell expresses only one taste receptor, we say that taste is encoded as a labeled line, at least in taste cells. That means no matter what you do, if you stimulate this taste cell, no matter what the stimulus is, it's going to be either umami or salty or whatever that taste cell is stimulated by. The best data on this comes from some mutant mice that are actually forced to express the bitter receptors in sweet taste cells. So by doing a little bit of genetic manipulation, they've caused the sweet taste cells to now be activated by bitter compounds. Naturally, essentially all animals like sweet. And there are very few who naturally like bitter tastes. So bitter is generally seen as bad and off-putting, okay? Whereas sweet is seen as pleasant. And so mice, much like people, naturally like sweet things. And so the preference for mice, if given it, is to drink sweet water. They certainly won't like bitter water. So if we look at these data shown on top here, I want you to focus on 2 of the trends. There's the red and the blue. Now what we're looking at is a preference for water that has a bitter taste in it versus normal water. Usually, mice should dislike bitter water, and in fact, when you express the bitter receptor in bitter cells, which should be done normally, you see that. So look at the blue line there. You'll see as you increase the concentration of the bitter tastant, the mice prefer that bitter water far less. On the other hand, when the bitter receptor is expressed in sweet cells, now the mice actually prefer the bitter tastant. So in that red trend, what you can see is that there's an increase in the preference as you increase the concentration of the bitter tastant. In other words, it's no longer bitter to the mice. It seems sweet to them because they prefer it. And that's because the sweet cells, which send information to say this is sweet, they now express bitter receptors. So the bitter compounds are able to activate sweet cells, and thus they are perceived as being sweet. So the actual taste cell that gets activated seems to be really the determining factor in what are we going to taste. Once we have activated our taste receptors, we're then going to send that information on up into our cortex via the gustatory nerve. Okay, so ATP stimulates the gustatory nerve there in our tongue. That's going to be sent into the medulla, into what we call the nucleus of the solitary tract. Then it's going to go on up to the thalamus, in this case the ventral posterior medial nucleus in the thalamus, and then it will head on over to the gustatory cortex. Now, we don't have maps like we do in our somatosensory cortex or in our auditory cortex. Instead, we kind of have blobs. There's definitely organization, and similar tastes tend to be grouped near one another in different people, but you can see taste is very individual. Here we have taste maps for 6 different individuals. So all those 6 colored diagrams are showing you which area of the gustatory cortex was activated when the person tasted something sour in green, bitter in pink, salty in blue, sweet in red, and umami in yellow. And you can see that while it is true that the colors tend to clump together in each person, although not always, there's not a consistent pattern between each individual. And that's probably because of the relationship between different tastes. It's easy to say the relationship between our finger and our palms, and our palms and our arms, but what's the relationship between sour and salty? I don't know. So there isn't really a clear way of mapping this, and so it becomes very individualized in each person. All right, our last sense, olfaction. This is in some ways a little simpler than taste because there's no ionotropic receptors, but in the end it's very, very similar to how we detect taste, except we have to detect many, many different molecules because there are many different odorants. We can do this because we have a very large family of G protein coupled receptors called the odorant receptor gene family. Depending on what species you're in, you will have hundreds or thousands of different odorant receptors expressed. These odorant receptors are going to be found in little ciliated ends. So again, we have those little tiny projections coming out of, in this case, olfactory sensory neurons. All right, so if we have a look in this illustration, what we're seeing is somebody sniffing a flower of some sort. You'll have to excuse me, I don't know my flowers. And if we were to zoom in and the upper part of their nasal passage, what we'd see is their olfactory bulb shown in yellow on top. Then there's this little projection that must head through the bony covering and hit the olfactory epithelium. That's where our olfactory sensory neurons are embedded, and then exposed in our nasal passages. You can see these little tiny cilia that are littered with G protein coupled receptors. Those are on the bottom. So they're exposed to air. So as we breathe in, the odorants in the air are going to come in contact with these different receptors. And the many different receptors there will detect either a broad class of odorants or one very specific odorant. in the end will be activating olfactory sensory neurons, and they're going to then project back to the central nervous system. The first stop along the way would be the glomeruli. You can see them in this illustration. They're those little balls where all the green, blue, and red olfactory sensory neurons project to. And we do see these little balls when we look for them in real life. On the upper left portion of this illustration, you're seeing a glomerulus. Now, it might look like that's a cell body and it has some dendrites coming out, but it's not. There is no cell body here. These are the axons from the olfactory sensory neurons. They're the dendrites of that projection neuron that they're projecting to, and then there's some local processes of glia and local interneurons. The cartoon version is shown below, so on the bottom left we can see the different glomeruli. So these regions in the olfactory bulb, this is where olfactory sensory neurons that detect the same type of odorant send their axons. They all converge on the glomerulus. All right, if you want to see the real-life illustration of a glomerulus, you can see it on the right-hand portion of this illustration. On top, you can see the olfactory epithelium. That's where the olfactory sensory neurons have their little ciliated projections. Then they project to the right into the olfactory bulb, and you can see at the very end, there's the glomerulus. From a different view, you can see on the bottom where there's a few different glomeruli shown. We really don't know how the axons find their way to this specific glomeruli, but it's clear that it depends on the odorant receptors expressed in the different olfactory sensory neurons. If you were to knock out specific olfactory receptors, you see the loss of certain glomeruli. So we can see our somewhat normal mouse on the bottom. You can see very clear glomerulus showing up. On the top, we've deleted one of those olfactory receptors, and you can see that the neurons that should express this but now lack it, they're illustrated in kind of that bluish green color. They don't form a glomerulus. They can't find their way there. Again, this guidance mechanism is very poorly understood, so it's not something we're going to talk about in this class. But in all likelihood, the receptors get activated, and then they stimulate intracellular signaling pathways that then modify the cytoskeleton, so that the neurons that have the same receptor have their cytoskeletons kind of coordinated in a similar fashion, so they meet up in the same location in the olfactory bulb. Beyond that, it's still somewhat mysterious. From the olfactory bulb, we are going to project directly into the cerebrum because the olfactory bulb is immediately below our cerebrum. It doesn't need to go through the thalamus. Instead, it's going to go directly to the amygdala, where it's going to the emotionality of the smell will be determined, so the pleasantness of it, the memories will be determined by going to the hippocampus, and then there will be a projection to the orbital frontal cortex, where we'll become aware of these smells. Just like tastes don't have a real map, if you will, neither do smells. They have a sort of rough zones where like smells are grouped, but there will be multiple zones for the same types of smells. So there's not a clear map here. And again, what's the relationship between one smell and another? There's not a clear spatial relationship there, so it's difficult to map that spatially. And there we have it. So there's our smell system right there. We pick it up in the nasal passage with those G protein coupled receptors. Those stimulate olfactory sensory neurons that then send that information up to one glomerulus or another. The projection neurons there in the olfactory bulb pick up this signal and then relay it to our cerebrum, where we can determine, have we smelled this before? Do I like it? Does it bring up any fond memories? And that does it for all the other senses. If you have any questions on this material, don't hesitate to email me, and I look forward to seeing you in our next lecture.